Hello everyone. In our today's lecture of genetics, we are going to learn about the tetrad analysis. Now, in certain species of the lower eukaryotes, such as algae and fungi, which spend most of their life cycle in the haploid state, also have used the mapping studies. Fungi can also reproduce sexually and produces four sexual spores. As it produces four such sexual spores, which are haploid, so they are known as tetrad, and the tetrad formation takes place. And in our today's lecture, we are going to study about these analysis of those tetrads. In some, meiosis is also followed by mitosis to form 8 spores known as octads. There are different types of organisms which are used for the study of tetrad analysis. For example, lower eukaryotes such as algae and fungi, especially fungi like Saccharomyces cerevisiae, Cornipus, Lagopus, Chlamydomonas, Reinhardt tetrads, and Neurospora crassa or Aspergillus nudulans are used in large scale or extensively for the tetrad analysis. Now, tetrad analysis in some species like fungi, each meiotic tetrad contains a sac-like structure. In that sac-like structure, the four sexual spores are present, and that sac-like structure is known as ascus. In the diagram you can see this is the sac-like structure which consists of tetrad, and this sac-like structure or ascospore containing structure is known as ascus. Each product of meiosis is an ascospore. You can see in diagram, in ascus a single circular structure, or each product of meiosis is known as ascospore, and it is formed from one meiotic cell remain together in the ascus. Then in the tetrad, when two pairs of alleles segregates, the three pattern of segregation are possible. Means whenever the crossing over or the segregation of tetrad takes place between the two pairs, and then uh, three types of patterns of segregations are possible, in which the first pattern is parental type. In the parental type, two parental genotype segregates to form the uh, segregating pattern of tetrads. Second type is non-parental type, which is also known as NDP, and here only the recombination process takes place. And the third type is tetra type or TT, in which all the four types of genotypes are observed. See, here you can see the ascus structure, in which first diagram is of your parental type. Tetrad formation takes place, in which only two parental type ascospores are observed. Then in the second diagram you can see the non-parental type tetrad, and the last structure is of your tetra type tetrad, in which all the four types of ascospores are observed. Then there may be orderly arrangement of tetrads, or there also be unordered tetrads can also be observed. First we'll study the analysis of ordered tetrads. Here the tight ascus is produced, which prevents the spores from randomly moving around. In the orderly tetrad, the ascospores does not move from their location. They are tightly adhere in the ascus. For example, in the fungi Mucospora crassa, the ordered tetrad is observed, and the map distance observed in it is 1/2 number of Asst, which is your tetra divided by total number of asci into 100. This is the formula to count your map distance. The formula of map distance is 1/2 into number of tetra type, sorry, 1/2 into number of tetra type divided by total number of tetrads into 100. By putting all the values in this formula, we can count the map distance between the two genes. Here the example of Mucospora ordered tetrad is given over, in which the ordered asci can also be classified as parental ditype or non-parental ditype or tetratype with respect to two pairs of alleles, which makes it possible to assess the degree of linkage between the genes. This we can study with the help of one more example. Now, how does this tetrad analysis takes place in the ordered tetrad? First, the homologous centromere of the parental chromosome separates at the first meiotic division. Whenever the first meiotic division takes place, in the first meiotic division, the homologous centromere gets separated. After the separation, the centromeres of the sister chromatids separate at the second meiotic division. पहिल्या meiotic division मध्ये काय होतं की जे पण homologous centromeres आहे, पॅरेंटल क्रोमोझोम मधनं ते homologous centromeres सेपरेट होतात. ते सेपरेट झाल्यानंतर सेकंड meiotic division मध्ये सिस्टर क्रोमॅटिड्स एकमेकांपासून काय होतात, सेपरेट होतात. Then in the next step, when there is no crossover between the genes and the centromere, the allele segregate in the meiosis first. Then the allele segregation takes place, and at the last step, the crossover between the genes and the centromere delays the segregation alleles until meiosis second. मग meiosis second येईपर्यंत जीन्स आणि सेंट्रोमियर मध्ये काय होतं, क्रॉसिंग ओव्हर टेक्स प्लेस. सी हियर यू कॅन सी दॅट इट विथ द हेल्प ऑफ एक्झाम्पल. See here you can see the homologous centromere of the parental chromosome. Here the first division of the segregation takes place, where the no crossing over between the genes and the centromere occurs in the first step. Then at the meiosis 1, the homologous centromeres get separated. After separation, the meiosis second takes place, and each sister chromatids सेपरेट फ्रॉम इच अदर. आता तुम्ही इथे बघू शकता की सिस्टर क्रोमॅटिड्स पण काय झाले, सेकंड मियोसिस स्टेपमध्ये सेंट्रोमियरपासून त्यांचं स्प्लिटिंग होऊन त्या दोघी पण काय झाल्यात, सेपरेट झालेत. देन द मायटोसिस प्रोसेस अकर and the possible spore pattern with the first division segregates. Then any of the type of segregation may takes place. हिअर यू कॅन सी इन द डायग्राम. कुठेही त्यांचं काय होऊ शकतं, जॉईनिंग किंवा सेग्रिगेशन त्या दोन्ही स्पोर पॅटर्नमध्ये फॉर्म होऊ शकतो. देन at the last step, possible spore pattern with the second division of segregation takes place. Then at the second level of segregation, any of these four possible segregation can takes place. या चार पैकी कुठल्याही टाईपमध्ये ते जीन्सचं काय होऊ शकतं किंवा अलील्सचं अरेंजमेंट होणं शक्य होतं. अँड दिस होल प्रोसेस इज carried out with the help of ordered tetrad analysis. Then the next one is analysis of unordered tetrad. Here the ascus provide enough space for the tetrads to randomly mix together. आता इथे काय होतं, आपण ऑर्डरमध्ये बघितलं होतं की तिथे जे काही टेट्रॅड्स किंवा ॲस्कसमध्ये जागा नव्हती, सगळं इंटॅक्ट असल्यामुळे ॲस्कोस्पोर्स त्यांची जागामधनं मूव्ह होऊ शकत नव्हते. बट अनऑर्डरमध्ये टेट्रॅड काय करतो की ॲस्कसमध्ये त्या टेट्रॅड्सला मूव्ह होण्यासाठी इनफ स्पेस असते, त्यामुळे त्यांची ऑर्डरली अरेंजमेंट होणं पॉसिबल होत नाही. तिथे अनऑर्डर्ड टेट्रॅडचं काय होतं, अरेंजमेंट होते. अँड दिस सच टाईप ऑफ अनऑर्डर्ड टेट्रॅड इज ऑब्झर्व्ड इन Saccharomyces cerevisiae. Then what is the advantage of this tetrad analysis? First, it potential for analyzing all of the product from each mitotic division. मायटोटिक डिव्हिजनमध्ये जे काही प्रोडक्ट फॉर्म होतं त्यांचं पोटेन्शियल चेक करण्यासाठी आपण काय करू शकतो, टेट्राड ॲनालिसिस यूज करू शकतो. देन हॅप्लॉइड इन नेचर, produce very large number of progeny. Then the life cycle of these organisms tends to be short, can make direct observation on the behavior of the genes during meiosis, and can examine crossover. And last one is can map the centromeres. These are the advantages of tetrad formation. This was all about the tetrad analysis. Thank you.